bit mixed up. Sometimes it's even hard to tell what is the axon and what is a dendrite, but generally this is the simpler idea. So, now that many of you are engineered like precision, we can start doing a kind of like a block diagram, right. So, here is a processor. So, let us, and then you see how the inputs are coming. So, let us draw a block diagram, cell body, lots of input is coming. Yeah. Now, you can see one of you asked question about do dendrites connect to each other. Yes, you see this dendrite is connected to this dendrite which is connected to the cell. In fact, here one is connected here, one is connected there. So, what is happening is that this particular dendrite input which is coming from some another neuron is sending a message which is modulating this input. It can block or it inhibit. Always synaptic connection can be excitatory or inhibitory. So, imagine if this is inhibitory, then if the message was coming here to the cell body, this will provide a break, it will inhibit it. So, that is the kind of message here, or it can excite and reinforce it. So, suppose I see your face and I hear your voice, and let us say this is coming from my eye and this is come from here ear. If the two happen together, likelihood of me recognizing is higher because it reinforced the learning. So, that is the kind of way that this, I mean this is of course I describe it a very macro scale, in reality it is microscopic. In this one is closer to cell body, this one is further. So, you can imagine that this one is more weight, more emphasis, because this guy can block or modulate this message. So, you know a lot of these kind of summation is occurring, weighted summation. Do you get it? It is weight. How much weight of this, how much weight of this, sum it up. If the weight is strong enough, it will affect the cell body. Now, cell body sitting here is like an integrator, it is integrating this, this, this. Then it decides based on everything coming at any time, is this important enough, then let me send the message out. So, you can almost begin a emerging model. So, as I said, their inputs are excitatory or inhibitory, and you can have this one is an excitatory synapse, this is an inhibitory synapse, this one is a little bit of both. Yeah. And how is the message being given? Message is always through action potentials or spikes, which means these message is coming in the form of this spike pattern. Note that it is kind of random, it is not precise, there is a stochastic is what we would use, it is relative random looking. People who are studying telephones, internet trafficking looks the same. There is no way to know. I mean, we know that internet traffic might go up during the day, might reduce during the night. Well, EEG changes during the day and night, we know that. But if you look at somebody sitting on an internet switch, the message is coming, it will be random, right. So, it is the same way that there is this random, but there is a message there, there is a code there. And when this information is summed up, the cell will react. So, how do I take advantage of it? How do I put it in silicon? I mean, can I make an artificial circuit out of it to explain this? Yes, you can. So, you do not need to know this circuit, but this is what somebody has actually built, a detailed circuit, which means there are some excitatory synapse, there is a membrane potential, there is some model of a N channel, there is an inhibitory synapse. So, this is like a neuron in a silicon electronic circuit. So, hardcore electrical engineers designed this kind of thing to make it look like a neuron. Anyway, let us come back to biology. So, this guy Raymond Cajal was one of the most famous neuroanatomists. He was the first person who utilized this staining technique called Golgi stain. I think, okay. So, Golgi invented this something out of silver, you know, like you guys are very young, so you do not even know the regular old fashioned photograph, but you know before the digital photography there was a standard Kodak and all that was print photography, and it was based on chemicals that would take light signals and change the chemical, and then you suddenly change color and becomes a print. And so, silver has that kind of reactive effect to change color, and he used that kind of silver like stain, and now you can see that it stained the brain cells into black and black. Remarkably, it does not stain all the cells, just few, which is really lucky, because it stained everything, this would be so dense we won't see anything. But luckily just few arbitrary cells are stained. So now you can see at a slightly lower density that there are these different types of cells here. These are what are called pyramidal cells. They are some of the biggest cells in the brain, and they have a huge network of dendrites. So here you can see its axon, and then there is a large network of dendrites that go and travel. Many remain nearby and connect to each other and come down and synapse here, but many go far away and connect message. These are what are called interneurons. These are neurons sort of in between, and you can see that they are modulating kind of like this kind of an effect. They are kind of locally doing it. That is why I answer to your question about can it go back and do it itself? Maybe, but extremely difficult to follow. This is real image, but huge simplification, because probably in this case 1 out of 1000 neurons is visible. So if there were all others, this would be so dense we wouldn't even know, you know. And so it's kind of like, imagine a train station in Calcutta train station on a peak hour. You can't tell in the mass of humanity, but when in the night or evening when the density goes down, now you can tell, oh, these are different people, different station, maybe you look at it, you can find that. So it's lucky that stain became like this, and now we can find the pyramidal neuron, interneuron, and from this we started to build a bit of architecture that there is input, there is output, there is a local control and feedback provided by these guys. So this is one way to look at it, but this is actually at a much higher resolution way to look at it. Now when you look at it at a very high resolution, again roughly get an idea that this is about 10 microns, so it gives you some idea, and so you can see that these dendrites are about 1 to 2 microns, sometimes more. But then you see these tiny blue dots, and these are the stain with which you can tell where the synapses are. So these are synaptic buton. It's a little tiny bump in each of those. So every one of these is a bit of a bump, not just blue, but some of the yellow bumps also. So you are not, because again none of these stains stain everything. So there is some other neuron again imagine this is packed 3 dimension, but luckily because it is not entirely visible, now you can do a little bit of tracing that, hey, something like this came and looks like it made a synapse here. So that is what you can, if you are lucky, see under the microscope, and that is from which you have to figure out how the brain works, you know. So it is very, very challenging. Okay. So let us go back to elements of the neuron. Now it is a little bit more detail. Again, I cannot imagine you can remember, memorize, although people who are biology or medical students probably have to memorize all this. So what is inside? Well, inside is the nucleus, and then there are these substructure. Again, don't worry if you can't memorize everything, but try to remember as much as you can. So of course it's made up of what's called cytoplasm. That's all the fluid inside the cell. It's kind of like a bag of fluid, but it has a nucleus, and it has what's called nucleolus, but it has a very critical member called mitochondria. That's the energy store. These things, these little egg-like thing you see, are the energy store, the energy producing part. This is where oxygen produces reaction to provide ATP, which is a very high energy molecule. So that it's the energy. The so called microtubule filament is all structure. They give it a structure, like it becomes a physical, because remember it's like a balloon, a bag of water. So what gives it structure? So these microtubules and mitochondria and everything else give it a structure. Then I've already told you what is myelin. So it's like a sheath, although it turns out it's not continuous. So you can see it's bump, 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 bump. So it kind of like electrical activity goes from here to boom, to boom, to boom, you know. So it kind of jumps, and that's why it goes very fast. So anytime a axon is myelinated, it makes for a fast transmission. And because of some disease, when you lose the myelination, MS and those kind of diseases, certain motor functions gets impaired. Yeah.
So the gaps between those cells, they are the nodes of triangular.
Yes. Yes.
So they are not myelin. They don't have myelin.
Again, this is a very simplistic cartoon, but there is a gap, and that allows for external electrical field from here to there. So there is some structural gap. the myelin, it looks cute like this. In reality, it's kind of like another cell. There's certain cells called Schwann cells. They kind of wrap around, and they're also all three-dimensional. So I think this is a nice artistic simplification. Okay.
So my doubt is, like, if the myelin sheet gets some breaks in between, why should that mean that the transmission goes slower?
Well, I mean, so there's a lot of electrical engineers, others have done a tremendous amount of physical modeling and experimentation to it. So without going into a lot of theory, it's called kind of cable theory, it's called. So electrical cable information proceeds the same way. So this is very structured to the extent that you have these kind of insulation and gap. It's called what is called saltatory conduction. It can happen. But when you have a degeneration, it's never simple like this cell is dead. It's all, there's a lot of breakdown. So the flow is never not now as conducive. So basically it changes it's not like on and off, but it messes up the conduction. So the whole timing is off, right? If you're a cricket player and a little bit timing is off, suddenly that person, superstar, next day cannot play well. Very small timing difference. And all of a sudden, right, playing this way and a little timing difference and it's a catch. So, things you have to understand in that level of approximation. Okay. Yeah.
Is there any research going on in
everywhere, all the time, in all the areas?
Like electrical engineers using creating artificial myelin sheets, that kind of thing?
No, there are two separate questions. Electrical engineers have spent a lot of time. It was very fashionable at one time to build electrical model, that's called network model. There was a very high analogy the same way that electrical wiring, electrical messaging occurs. The same theory is applicable because they are resistors, capacitors. They model this whole conduction by resistor, capacitor, inductors as a way of conduction. It was very popular for a while. It's no longer as popular. There are a lot of biomedical and biological and biomedical engineering researchers who are studying how to remyelinate, right? The demyelination occurs, of course, we need to know why. Probably some kind of autoimmune or some reason why that damage occurs. So how can you prevent it? So there is a lot more research in what is called tissue engineering is a field, regenerative engineering, where you make these things regenerate. Okay. So as I said, there are lots of different types of synapse. This is a synapse. These are synapse exodendrite. So axon to dendrite synapse, synapse axon to axon synapse. So everything is possible. What does a synapse look like? What's inside a synapse? So that's the magic point, right? So remember, like I told you in that micrograph, those little bumps where we saw these little bumps, these little tiny bright spots. That's what these are. These bumps is where the action occurs. So what is it? So this is what we would call presynaptic, like the input part, and this is the postsynaptic output part. And it releases small amount of chemical, neurochemical it's called. It's usually stored in little tiny like bubbles. And when the message has to go out, when an electrical message comes, then an electrical message comes and tells the bubble to migrate here to this point. And then the bubble, it's like it's lipid, it's like soap film. The bubble fuses and opens up this and the chemical pops out. When that chemical pops out, it wants to then find a target. So on the other side is the receptor, which has all these molecules of proteins that perfectly look like target, like receptor. So they're ready to receive these tiny little quanta, little bubbles. And then the action potential that came here, which initiated a cascade of these little bubbles, which in turn produce little tiny dots, which in turn go and connect here. And when they connect here, this cell says, yep, I've received a message. So it's a kind of like electrochemical message. Everyone understand, right? Electrical message becomes a chemical message, chemical here, back to electrical. And so those chemicals are the neurotransmitters. So when neurotransmitters, something breaks down at any level, presynaptic, postsynaptic, too much chemical, too little chemical, drug addiction and all that can happen here because it's, these receptors are unique to a particular chemical, and that chemical can be addictive. So addiction is based on the fact that these receptors want that chemical. If they don't get it, they send the message, I want the chemical, give it to me, So, there are this kind of a lot of factors, but because if we are interested in learning, memory learning, that is happening here, that if you use more of it, there will be more of these, this connection will be stronger. It will take less of these little tiny dots to make a output. Okay?
Sir,
Yeah.
there is a gap between the presynaptic
Yeah.
layer and the post, that is called synaptic cleft.
Mm-hmm.
So the neurotransmitters are not directly binding the receptor.
No.
Some of them are staying in the synaptic cleft.
Well, they diffuse. They are not staying because they are recycled very quickly, because that is a very high energy. This is where I think brain burns, I don't know, 30-35% of your energy of the whole body. Most of the energy goes here.
So is the synaptic cleft is having the role of memory?
Of what?
Memory. Because
Well, it is not so much the cleft, the whole thing, right? I mean, what is memory? Remember, again, I have tried to, memory has many factors, but there is nothing called a number here. It is not a memory like number 1 is here. What it is that if I send you a message, if the message is familiar, more of these little bubbles will be sent out and your result will be stronger. If I do not have that, the result will be weaker. That is one kind of thing, right, because it is reinforced. The other kind would be that this is sitting there, if the same message comes, you will give the same message, no big deal, but a new message comes, yes, surprise, big result. So, there are all these type of this. So, the memory in itself is nothing one point, there are trillions of these synapses. So, the memory is shared in all of these things.
So what I read in the literature that at the postsynaptic membrane we have some few amount of receptors and lot of neurotransmitters are coming for binding, but they cannot bind because very few receptors are at the postsynaptic side.
Okay, I think so. We are going, see, because if I go down, I mean, again, this is like a whole course in synaptic learning and memory, entire semesters. We will get so microscopic that we will lose the broader perspective. Will be here long term. I think let us keep it at a more general principle. I mean, these kind of things, first of all, also I can't say I am expert on an entire, because molecular machinery, here there are dozens of proteins involved, dozens of genes involved. This little tiny picture, this is sort of like high, reasonably high level to give us an understanding of how these things work. But for molecular biologists, they could be studying hundreds of proteins, chemical reactions, closed loop. See, I am already seeing some alpha waves and theta waves creeping up, because this is going to fall asleep. Wake him up, his memory is his synaptic cleft has opened up, it's so let's move at a level where higher, and then when you have very fine questions, I don't mean to be rude, but we can take it up offline and discuss that. Okay. So you see, the neuron is, oh my god, so much detail, receptor, neurotransmitter, neurofibrils and microtubules. Microtubules are also structural as well as they have role. Some people even think that is where a lot of the intelligence lies, in the axon, not in the synapse. That is another theory. So, too much to learn. Every time I think of the brain, I also look at space, right? How much, we all understand, right? How much do we know about what is out there, far away galaxies, far away black holes, even in our, right? When we have, well, our telescope is not good and it is okay. Well, think of the same way the brain, it is going to be the last frontier, and there is just going to be so much detail, and it is so complex. So, luckily we made some headway. So, you see how the myelin looks like, it is like a insulin wraparound. So, basically these kind of cells wrap around. So, yeah, everybody memorize now. See, so it is very difficult. Synapse, synaptic vesicles, external terminal, you can just go on and on. There are so many different types of molecules involved. You see the little mitochondria, the energy stores, nucleolus, ribosome, Golgi apparatus, these are all things inside the cell, lots of different types of synapses, fun. And even at that coarse level, you can see how messy it looks, and it is really 3-dimensional, highly packed. This is only because this lucky break of sparse staining. So, I think I will skip the table. This is, if I upload it somewhere, you guys can look at it in the details, too much detail. So good old Raman Kahal Said, yeah, neurons are many types. This is a Bengali, this is a Maharashtrian, this is a Punjabi, this is a Gujarati, this is from Kashmir, this is from Nepal, China, Japan. Neurons are of many different varieties. The most ones that people relate to, that earlier I had shown you, what's called Purkinje cells, it's the biggest, richest one. You know, this one thing is going to send millions of connection all over. It's in the certain part of the cortex. So it's more studied, but there are all of these. Some have, you know, this one is a what again it's called pyramidal, but it's just simple. It takes its output is here, and input is coming from all over. But others are messy. They are so complicated. They have all these shapes. We don't know how to figure this out. They might be called interneurons. So on. Okay. So neurons are multipolar, bipolar, unipolar. Unipolar 1 to 1, bipolar 1 this way, multipolar connecting to everybody. So if you want to make a structural study, you can do that way, right? So here is a Purkinje cells, output, input, very good. This one is simpler one, and these are what I call multipolar. Take lots of input, millions, tens of thousands, hundreds, output, input, output, like that. Neurons can be all over the place, so don't even just the cortex. They are in spinal cord. So spinal cord, remember at one time I told you that we'll look at spinal cord. See, this is what a spinal cord looks like. So inside this gray area is where all the spinal cord cells are, and then they send out this axon, what's called motor neuron, to the muscle. In the eye, you have what I call bipolar neurons. So they are between those rods and cones and everything, and then they send message to the brain. They are sensory neuron in the spinal cord. So information from the skin goes into the spinal cord, makes a synapse here, and then sends the message to the brain. Different types of neuron. I showed you this picture in the morning. This was only last 5 years ago or something. You know, Cahal did it in 1890, around 2000. I think he got the first Nobel Prize. But subsequently, there have been Golgi stain and a lot of other stain, and now this colorful way of, because they give this colorful proteins using virus. So they code different types of cells with different color. Do you see the density here? Wow. You know, and this is again sparse, okay? It's not capturing everything. Okay, everybody memorize the chemical formula. These are different types of neurotransmitters: GABA, epinephrine, norepinephrine, dopamine. You know, some of you know those names. See, this is slight depiction. How many synapses? Yeah, 10 to power 4 is kind of people easily throw around. This is what is called, so this is the, with a light microscope. Fluorescent microscope provides a color, and these little dots are, it's captured many of the synapses. This is just one of those neurons. You can see how many connections are there. Again, imagine in 3D, okay? We are just seeing the 2D part of it. So take it down. Remember, each of these, this circle may be about 10 microns. So this is about 2-3 micron, and some of these dendrites are about 2 micron. So now this little dot is this. So now this 2 micron cross section, and you can see the mitochondria here. They have this kind of like special pattern. Maybe one of this might be cell body somewhere or not. And this is the synapse. This is at nanoscale. So this little black coloring is between this dendrite and this axon or whatever, where there is an or cell body. Very hard to tell. By the way, this might be myelin, this thick covering. So this particular axon is myelinated. It has a thick covering. Do you see that there is no space? Remember I told you about extracellular space for Alzheimer's, yeah, or in sleep. There are some recent research that show that when you sleep, neurons shrink a little bit. They open up these little tiny extracellular space so that all the chemicals and debris of the day can be cleaned out and removed. Otherwise, look at the packing. This is what we are dealing with. Yeah. So just sit there and enjoy awe of what this is. So here is the synaptic cleft, neurotransmitters, chemicals coming from here to here, receptors. this is what I told you, it gets so complicated. You know, this is the more detailed analysis. This was prepared by one of my postdoc, and his entire PhD was on this. Think about it, right? So, as a student, you may have studied all of this, but synaptic. So, message comes, goes to the precursor, this, that, then endosome. They recycle some of these, because you have to recycle everything. You know, the whatever comes back has to be recycled and brought back. Otherwise, where would it go? All right. He got a Nobel Prize for some of these detailed works on neurotransmitter release and very precise microscopic. Here. Okay. What are the other types of cells? So, right. So, let's go back to the brain. Brain is three-dimensional. This is all this medical language. Okay. Superior, or it's called dorsal. Inferior or lower, or called ventral. So, this is the top of the brain. This is the bottom of the brain. This is the small brain, cerebellum. This is the large brain, cerebrum. This is the prefrontal cortex, with a lot of brain, a lot of intelligence. Temporal lobe, a lot of speech and language somewhere here, vision somewhere here. The motor cortex is here. So, these are crude you can see the gray matter, which is where brain cells are. White matters are axons and dendrites. Okay, so it's a lot of details. I've shown you this. Why are axons myelinated? What would happen if we didn't have myelin? You know, message would be slowed down, it will be messed up. You get multiple sclerosis. As I told you, what really is happening is that when you think of that myelin, it's like this particular type of cell. It's called oligodendrocyte. It's a cell, and it sends out legs which has part of the cell, and then they wrap around like this. Again, all of this in 3 dimension. Okay, we are just giving a nice simple picture, and you know, so these are details of conduction. So you've seen this. Oh, this is the nerve, right? So think of nerve. You say, okay, nerve is axon. No, it's not. It's too complicated. This is the nerve. It is to protect it as a sheath. So it's called epineurium. It's a sheath around the inside what are called fascicles, which is another. So it's like a big cable. Within that, a smaller cable. Within that are these fibers. These are the nerve fibers, and of which some might be myelinated and some may not be myelinated. By the way, there are always blood vessels in there, because even nerves need oxygen, need nutrients. So you can see in a cross section it looks complicated like this. That's the nerve. And by the way, some things can carry sensory message and some can carry motor message. It's all mixed up. So there are different diameters, as I told you. They can go from 0.1 micron to 20 micron. Very, very so-called C fiber, very small. They carry pain, touch, and other information. Pain is very fast, so you need to get it very quickly. And these are slow movement related fibers. So there are many different types of fibers. So if somebody, this is now a symbolic example, that if you have a nerve and it gets damaged, what can happen? You know, so surgeons will suture up the nerve. They can do that. People who are bionic would make a connection from here to there to make it work, right? So this is somewhere, if the nerve is damaged, you create an electronic device that will record and send the message on this side. I'll show you some videos later. So this is one of the projects we started in Singapore, to build this kind of stimulation device so that you can record and then stimulate, or take the sensory information and send it back. Okay, so I think this is end of the chapter or presentation. So let's take a pause, stretch, yoga, some deep breathing, mindfulness. Think about the brain and your myelination. Slow down, alpha wave, theta waves. Mitochondria, where the energy is it going to the stomach nervous system or to the brain? Ah, he's awake. Yes, go ahead.
It could be a silly thing, but
no, please.
Neuron think of its own?
Sorry?
Can a neuron think of its own?
But what is think?
Think in the sense that certain functions.
There is nothing called think. There is no think. So I cannot let you go beyond that. because a think or a thought requires great philosophers to tell you what they mean. So, there is nothing called think. So, let us just move, it is so far out, it is like the professor before talked about emotion and I had to cut it off. We do not know, I mean these are philosophical and linguistic words, there is nothing called think. A neuron is not thinking, neuron is a bag of water with energy source like little batteries of mitochondria who allows neurotransmitters to go from A to B, presynaptic to postsynaptic. It is not thinking, it is just a chemical, it is a bag of chemicals sending messages. So, there is no point in saying the think. I would not even bother to answer the question, does a brain think? I mean, what does think mean, right? So, we get very off topic, and please, I am very serious, because I think we have I mean I understand you want to know about thought and feeling and emotions, but other than casual answers, there are no rigorous answers. I can give a casual answer. So, what does think means? It is a bunch of messages.
Relevance over here for this question. Then where does this variation rise for this various stimulations or EEG signals?
Everywhere, in every single point. The action potential spiking, the neurotransmitter release is stochastic or random, gene expression is stochasticity everywhere. This because it is kind of like if you look at thermal energy kind of thing, it's always there's some built-in randomness to it, and it's for a reason, because there's nothing precise about it. It's the collective lots and lots of neurotransmitter bundle up together to produce enough neurotransmitter to produce sufficient action potential to be an output. And one of the things that is hard for us to understand, well, okay, who is the physicist in the room? There are a couple of you, right? Quantum mechanics is hard for us to understand, right, compared to Newtonian. And what it is rather hard to action at a distance. They are all kind of weird phenomena at quantum mechanics, but it's what it is. That's microscopic scale. That's how nature behaves. I wish it was very simple Newtonian, but it's not. And I know you're a medical person, so you don't know what I'm talking about, but that's what it is. That quantum mechanics has that same randomness that neurons have at this scale. Some people make analogies like that too. So it's a collective action. So a lot of action of many things together makes. So that's why try to develop intuition. Don't get buried in the details. The intuition I told you about, that if I want to pick up a cup, our mind would be, if I were building a robot, I would have a robot looking at the camera and get a precise calculation and go there. What is the price I pay? It knows only that doing well. It cannot adjust this. Oops, I'm in trouble. These randomness allows it to create a vector. So all the neurons together will pull up and say, oh, it's a little bit like this. Okay, let's go and adjust our angle. So that randomness builds that degree of plasticity and adaptability to changes, because everything changes. Nothing, never my arm motion, I play cricket, never the same. Is it ever? Is that the fun part of it, right? If it was exact, then the batsman will figure it out. You'll always be gone. But everything is a little bit different, and there's a beauty in that. So they change it. So it's how the learning occurs. It's everything is this small randomness. Yeah.
So, when you place this electrode in the brain, then as we learned just now that it is all chemicals which is really stuff, then how does it get converted into an electrical signal?
Yeah. So the electrode put in the brain is generally speaking sitting outside in that bag of where all the cells were, and it's picking up the electrical field of the brain cells. It's not at the chemical scale, is nanoscale. Those synapses and all that I showed, versus electrode is 1 to 100 microns. So it's a micron scale. So it's sitting and picking up the field. That's why I gave you the microphone analogy. That if I put a microphone here, it'll pick up sound of few of you, as long as too many of you are not talking at once. And then I can tell, oh, you're talking or you're talking. So electrode is picking up signals from all the neurons in nearby. If they don't talk, fire all together, I pick up some signature. So think in this of analogies that you can understand. Okay? So think of neurons like you guys are. It's a little too orderly, so don't imagine the real brain is like this, but you're all sitting in this room row by row, which is stupid. It should have been chaotic. Then we would have also, our discussion would be fun. See, right now, you see how orderly we are? So front row says something, back row says something, it's very orderly. But if you organize like that, it will be more interactive, more fun. So think of the brain cells in 3D, and I put a microphone here and some sound comes up, which is you as a neuron spoke up, and my microphone is picking it up and transmitting the message to me, which I pick up. But if 4 of you speak up, now I have to make some analysis and make a sense out of it. And nervous system is working like that, you know. Actually, these neurons in the brain, when they put micro, they're not very active. They're usually, but there's so many of them and so much is going on. Yes.
So a neuron, by changing its threshold, is either more conducive to change or less conducive.
Yeah.
So is it like his answer, like a neuron thinking?
No. I don't want to use the word think. No. Let's use biologically appropriate word. It's like you can't impose your language on quantum mechanics people and say let's call it whatever. It's what it is. So neuron is not thinking. So let's not do that. What it is chemical thresholds. That's also a little bit step by step. What a threshold means, how many quanta of neurotransmitter it takes for the receptor on the other hand to say that I have received the message to produce an action potential. And that threshold, which means how many quanta it takes, can vary. If you use it too much, it can get overloaded, the energy to restore may not be there, and the neuron is quote unquote tired, it has utilized its energy to do all of that, may not respond. You guys have eaten too much, let's consider the analogy, you are not responding as quickly as I would like to be because you are a little lethargic. Replenish, good night's sleep, all the neurotransmitters, energy, everything has been restored, it will be more reactive. That is one way to look at that adaptability or restoration. Another way to look at is learning. So the more it is used, the more it is reinforced. In some cases, in other cases it might be inhibitory, so it might actually be blocked.
Yeah, what I read somewhere was that conducting through the synapse can change the RNA sequence.
Oh, okay. That we are going into scale. That's not even opened up because most of the people are not molecular biologists. So you're getting into finer detail. So unfortunately, I'm going to have to cut off some of those things because then that's what I'm saying. This is I showed you on some level, and many of you say, oh my God, what is all this? Too much mitochondria, Golgi, and this and that. Now, if you go from that to, and I just generally said proteins. Now, if you go in there and you talk RNA and RNA salads, you know, that's a deep molecular biology. It's not for this audience and this class. Yeah, yeah.
So
one by one.
The sizes that we talked here are in the scale of nanometers and micrometers. So this is the realm of quantum mechanics. So
not really. No. I made a kind of loose analogy to quantum mechanics because I wanted to convey that uncertainty is okay. Uncertainty has its meaning. So that's all I meant. But it is not at the quantum mechanics level because that is at atomic and subatomic level. So you know that we are talking about, when I say nano, in the sense that you then need that electron microscope. So you saw the circle was about 1 to 2 micron, and then synapse is that. But when you get down to a individual vesicle and there is a protein coming, the proteins are nanometer scale, but it is not quantum mechanics. Now protein itself is quantum mechanics because it is a bunch of atoms and molecules, right, atoms. So yeah, there is some protein folding is at quantum mechanics level, but not at the neuroscience level. You understand, every protein changes a little tiny structure, of course it has an effect on anything else. So yes, there is quantum mechanics at atomic scale, just like there is quantum mechanics going on in you right now, but at macro scale, right. Please, I hope I am not going too far and boring some of you, right. There is quantum mechanics in every one of your atoms, right, but there is a duality in quantum mechanics, right. So at a macro scale those disappear, we do not have to worry about it. So same thing in neuroscience. I mean, yes, there is quantum mechanics of at atomic scale, but at higher level it's averaged out, washed out. So let's not go there. You had a question? Yeah.
So you talked about long-term memory and short-term memory, right? So I wanted to ask, what if someone is doing a particular activity, over a period of time it becomes habit and gradually it will go to its long-term memory?
Yeah, I mean that's the very mysterious part of it, and what is the pathway by which that short term becomes long term. Like I gave the example of this Kendall and Schwartz who got Nobel Prize, or some of it was along that line, that they tried to study in these worms, and if they give tiny electric shock again and again and again. So worm gets a little reaction, right, it gets electric shock, so it reacts. but over and over and over, after a while it gets habituated. Then they do funny thing, right? Shock, 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 gap, shock, shock, and the worm is anticipating the shock. So it has learned that that shock is coming. So it has converted then into memory. With a rat or something, he goes in this maze, and every time he goes there, there is a food. Then he'll keep wanting to go look for there, even if the food isn't there, because it has got a memory. So it's converted its foraging into up to a point. After they will forget also. So the nice thing about all this is that we learn and forget, right? If we could, I think there are some movies like somebody who remembers everything. We'll go crazy, right? I mean, every gazillion, million, trillion information will be every single moment vivid. So we have to forget. So we only selectively remember some of the things. But some memories are stronger than others. So for example, a little baby rat goes there and it gets electric shock. That's going to get imprinted for rest of its life and never going to go there. Or you, when you're a little child and you ate something that was bitter, you never touch it again. You have so those are like, there's a part of the brain called amygdala, which is where deep, I won't call it memory, but these are emotions are attached to it, which is fear and those kind of things. So that fear memory, the feeling of sense of looking at a tiger or snake or something is so etched. It's a much deeper rooted memory, which is very different than the short term and long term. That's why I'm saying there are many, many types of memories. There are linguistic memory, there are spatial memory, there is a short term, long term is a simplification of, I give you my phone number 9234567, you remember it for a few minutes, after that you'll forget. So that's why we understand that, but reality there are so many memories. And so what we do is reinforce by repetition. But some things, what's called one-shot learning, like you just need to remember it one time and you'll never forget. So our brain and mind is mysterious. When you are younger, a lot more of those facilities are open. We used to be able to remember music, math, periodic math tables, all of that very quickly. Get older, that's gone.
An objective behind the saying that it takes 21 days to form a habit. This time has to be something with.
I think these are certain simplification, because that way people can somehow relate to it. They are not rules. They are not Newton's laws by remotest chance. It is cultural. It is a simple way that parents or society can pass on some message so that you can remember. You should do fasting once a month, or we like to impart some certain rules so that we create a good behavior pattern. But that 21 is utterly, completely meaningless. It's just a way to repeat. There was a very, very, very highly popular book. I don't remember the title. But basically, it's essentially came to the conclusion that to really become a world champion, something at that level, you need 10,000 hours. Essentially, they took like famous American basketball player Michael Jordan or some tennis player, they're a musician. They start learning at 4, they peak at 14. 10 years, 10,000 hours. It's a way to communicate that it takes incredible amount of practice and repetitions. There are people who would consider that the superior ingenious ability of meaning top tier athletic ability and so on, they are not innate, they are all learned. That incredible top tier athletes practice so intensely to the point where they excel and perform, and they are doing everything, learning, memory, practice, all of that. So yes, there is some truth in a general sense that repeat a lot, but in a specific sense, sometimes you just one time and you remember. Some people have precautious memory, right, of something they can remember huge number. I don't know how many numbers of letter pi that they can remember, like 1,000 of them. And those are, what you have to remember is these are this lot we don't know, and just accept it. Okay, don't seek simple answer and just be a good rational scientist. When you don't know, you don't know. In time, research and knowledge will catch up, and maybe we'll know a little better. I mean, for Galileo, with his knowledge and capability, trying to speculate black hole would have been meaningless, right? There was no telescope, there is no understanding. All barely he could make out was that the Earth and Moon and maybe planets are rotating. And look at how much trouble he had. So So we have to accept that we don't know what's in the black hole, what's in the galaxy, are there other civilizations, so the rest of it is speculation. But once the tools develop, now that we have better microscope, now we are beginning to see that there are other stars and other planets, and they are Earth-like planets. So now it's speculation that there is something there. But then the next better telescope starts telling us that there are in fact chemical signature in the spectrogram that says that there are certain organic molecule in that planet, in that star 50 light years from now. Okay, so we are moving in that direction. We have no way to know whether those molecules are relevant to life, and life in what form, so it's speculation, right. So I bring the space and telescope because sitting here you will say, yeah, sure, I can understand, I don't know. Well, apply the same thing to the brain. The level of details are all there, we just don't know, and accept it, and in time through meticulous research that information will come. You must understand the degree of complexity, right. So planetary motion was a very complex phenomena, right. Kepler and all that figured it out, right. But even then they cannot calculate precise trajectory of 3 planets, right. Those who have done physics, right. 3 bodies means unpredictably. So if that's the kind of uncertainty, so here also we cannot precisely model this, but we try to build simpler model like that. I gave you the summation network, it's called Hebbian network. So using that principle, people created neural network as a paradigm. And then using that principle for it took about 20 years of neural network until this shift of deep mining, deep network occurred, where they just said, why should we have 2 or 3 layers? Let's make it 30 layers and make it recurrent. And it sounded ridiculous, it's brute force, it's stupid. It's like saying there are 30 layers of brain networks, maybe, but there's no way to know, right? It's 3-dimensional. And so these, when the Google people did the deep mining 3D and gave millions of faces, and now they can do good face recognition. Nobody understands inside how it's happening. So those of you, everybody who has not heard of deep learning, everybody has heard about it, right? It's the magic, it's the machine learning, AI, it's the breakthrough of this, it's AI based thing, you know, beat the chess player or the Go player, all that thing. How? Go and it's a black box. Huge number of network, feed lots of every single game as input, and somewhere the little tiny weights are getting adjusted until it solves the problem. Same thing is happening. All these neurons, synapses are coming in and so on, and simpler rules have come step by step. Hebbian plasticity, weights are adjusted, use it or lose it, 21 days. I mean, these are simplistic rules of biology, because biology doesn't lend itself to equations like Newton's laws, at least yet. Although some things there are. Hodgkin and Huxley put it precisely in equation form, ion channel to voltage, which I think will be our next lecture. Okay? So don't get daunted by it, and let's take a few minutes, again 10 minutes break. But my point to you all is, enjoy the beauty and mystery of brain and all of that. And some of you may have a tendency to bury into the detail of an RNA and get into it, and if it's your interest, of course you need to follow that. Like I showed you, that PhD student worked on that little tiny circle in the entire scheme of things. So some of you may like that. Other people say, hey, you know, I want to build a brain machine interface robot, right? So it's nice to know the brain has certain organization rules and so on, and I'll keep that knowledge in my mind in a general way and make use of it. Clinical people say, oh, okay, I learned about this, and now if some of the engineering, how I can apply there. Some of the rest of you, oh, now I have some idea that grandma doesn't have Alzheimer's. Every time I say it, make them shut up. It's not, everything is not Alzheimer's. It sounds like it's probably sort of age-related dementia, which means it's really huge difference, because dementia is a slight progressive. It's like physically we get disabled, you know, we get weaker with age and all of that. Alzheimer's is utter progressive complete loss of memory because of tangles in memory. So don't condemn your family member by calling it Alzheimer's as soon as they forget something. A lot of people, oh, I can't remember that. Oh my God, do I have Alzheimer's? How many of you have Alzheimer's? Like, love to get it, you know, just to make it fashionable, because just you forgot something. Okay. Okay, let's take a break, and I think let's meet in this clock at 3:45. So that means you have about 12 minutes. Yeah, stretch, take a fresh air. May I suggest, saw the change. that you guys have rearranged yourself, which is I think good. That way, find another neighbor, talk to them about their interests, exchange ideas, it will be fun. Okay? Good. So it doesn't like this picture. Okay, good. All right. So the point here is that the cell that we saw in those electron micrographs or physiology and that Cahal and Golgi stain were structure. So let's give it a function. Let's give it a meaning. So how does a cell function? Now, for some of you it might be very basic. For some of you it will be first time. So one of the great things, and earlier in the day this mention was made of Hodgkin and Huxley. They got Nobel Prize for this, although any research was over the years was developed by a lot of different scientists. There are other Nobel Prizes for this topic also, because it's so fundamental that how does a cell function? And what I mean by function is in the context of its electrical activity, so that it produces for our neuron an action potential. So that if it has some input, it will produce output. How does it happen? And so basic idea is that the cell is made up of, it's like I've told you, a bag of salt solution. That's the simplest way to think about it, except it's got these little channels, what I call some proteins, that allow ions to go in and out. And when these ions go in and out, they change potential. So if you remember your chemistry, there's a chemical equation called Nernst potential, which when ions are different concentration inside and outside of a membrane, it produces a voltage. So of course, when the ions go in and out, the voltage will change. Ta-da! That's the mystery of an action potential. That idea is that using these channels, let ions go in and out to produce change in voltage. That's an action potential. So it turns out at the simplest level, the ions that are relevant are sodium and potassium. Lots of sodium outside, lots of potassium inside. But if you give an electrical stimulus, it flips. Potassium ions will be able to go out and sodium ions will be able to come in and depolarize or make the cell change its potential. So that's what this whole thing is about. A cell essentially is a cell, thing. This orange thing is what's called lipid bilayer. It's like think of it like a soap film. Think about it like a soap bubble, to be honest, but filled with water. But with little tiny holes at which something comes in. Hodgkin-Huxley's genius was that they predicted that for ions to go in and out, there must be channels. So when after decades of lots of real research, yes, they found that these are called lipid molecules. They act like this because the lipid layer molecules are hydrophobic, so they try to hide their tails here by facing each other, and the heads are here, and that's sort of like the double layer soap film in which these proteins are there, and then through these proteins ions can come through a rather energetic mechanism. It requires ATP, an energy source, and ions can go in and out. So in a way it's, and you see this cytoskeletal, you can say like everything gets complicated, but it's the structure. And so the issue here is that membranes like this can be permeable, which means allow ions to go in and out, even though lipids are very packed, but when the proteins are in there, things can go in and out. So what happens in the cells? Well, little sodium, lots of sodium, lots of potassium, little of this, and then they go back and forth. Other ions also involved, they play a role for sure. And as I told you about Nernst equation, what it says that if I took that lipid layer, that membrane, and separated it like this, it will produce a potential. So you can see that there is some potential between this side to that side. That's what Nernst showed. So here you can see that low molar sodium chloride and high molar sodium chloride, or vice versa potassium, there is a membrane potential. So this kind of membrane potentials is produced, but now let's do some magic. How about make it permeable to potassium ion to let it go through? Well, all of a sudden the equilibrium is lost. So if now, as it says, if the selective pore is present, this protein hole opens up and lets potassium go out. There will be a lot of positive charge here. Of course, something else can balance it, but then there is a change in potential and suddenly became plus 50 millivolt. Remember, it was minus, before that, and it goes up. So, point here is that these membrane potentials can change. You can have the other way around, sodium can go in this direction. So, this you can set up in a lab, in a test tube, or if you have a nice wonderful membrane, it is somehow ion selective, you can get ions to go in and out. So, let us go back to the cell. Lots of potassium, little bit of sodium, lots of sodium, little bit of potassium. You have a Nernst membrane potential usually around minus 70 millivolts. Okay, so that is where typical cell sits. That is the Nernst equation. So, Nernst equation says that depending on the logarithm, this ratio of the two ions, you will have a membrane potential. So, you could have with higher potassium one, and you could have lower potential and negative. So, the range of voltage now with potassium ion flow goes from 0 to minus 160. So, that is called EK, it is a, and this is the equilibrium potential, it will sit there. Okay. So, this is kind of like just for this clinical medical people, because they had the power in the morning, right, because they knew the language. So, now we give them the equations so that they can become more humble. This is now for the engineers. Well, so there is a lot of energy. This G is the Gibbs energy. So, and Nernst got a Nobel Prize. Wow, we will have to make a list of all the Nobel Prizes. But effectively, I derived this in the end, this equation, that voltage. I mean, these are some of the constant. It is slightly temperature dependent, charge dependent. F is Faraday's constant, but otherwise it is a logarithmic ratio of inside over outside. So, a voltage in such a membrane is dependent on inside or outside ion ratio, and then that can be applicable whether to potassium. So, again, outside or inside, this is the ratio, can be applicable to any ions. So, now let us go to neuron. So, how do you study neurons? So, this is the cell body. So, what we could do is to poke a microelectrode in the cell. These microelectrodes are typically kind of made up of little glass pipettes. They are, take a little tube of glass and then you heat it up and pull it, and it creates thin, sharp needle-like structure. Then you fill it up with some conductive solution and allows you to record. So, this is the record electrode, and this is a stimulate electrode. So, you can apply some current and inject stimulus. So, you see what happened was that we injected something. Now, I am trying to record the membrane potential, nothing. But when I applied a positive current, the membrane potential bumped in this direction. Okay? But something amazing happened at this point. When the applied current was higher, right, above certain level, all of a sudden this slowly rising potential suddenly become a spike, an action potential, just accelerated into like a pulse. That is the fundamental property of a neuron. It reacts to voltage above that injected current, or change in voltage from minus 65 to minus 50, which is its threshold, into a cascade of sharp change in potential. Because as soon as that happens, as we will see in next slides, very rapid exchange of sodium potassium current occurs, and suddenly the voltage shoots up to say plus 40 or something like that. And then as soon as it reaches that high, flips back and starts going back to 0 or even negative, and then there is a little bump like that, but in the end you get this action potential. It does show that if you give more, you do get some kind of different response, perhaps a little bit more and so on, but the main point is that you need to apply sufficient change in voltage, meaning you apply enough current that leads to change in voltage beyond the threshold to produce an action potential. So, that is the fundamental property of a neuron. Hodgkin and Huxley, they were doing all that in that 40s, and around that time, I mean, electronics were not like micro and all of that. So, they wanted a very long, very big axon, because this is too damn small, 2 micron is very small. So, they came across squid, which is a big fish, you know, you have heard about some giant squids and all that. Well, squids have somewhere in the body what's called giant squid axon. It is like millimeter or 2. It's like a tube. It's big. So in those days, they could insert a wire inside that axon. They could actually empty out what's inside and then shove a wire, because what's left is a membrane and channels. So using that squid axon is what they studied. Are you guys getting a hint on how to get a Nobel Prize? Find some strange creature somewhere, you know, worm, this, that. It's quite often, and then beat it to death, find some deep complicated thing, write some complicated equation, get a Nobel Prize. But you know, that's why you remember in the morning I showed you that strange weird looking axon that is going out of the way. That's where the breakthroughs are going to occur, because otherwise we keep on linearly thinking, right? Anyway, yeah.
On increasing voltage current, we can get multiple specs.
It's a kind of bit of a symbolic picture. It doesn't have to do with directly with increasing current. It has to do with excitability, and yes, it may, but it's kind of like, to be honest, it's a symbolic picture. So don't take it literally that if you have this much, you get this many spikes. Every cell is different, their depolarization properties are different. This is a bit schematic, okay? So this is a bit more Hodgkin-Huxley like. So now a little bit nice, this is more precise. So what they then see that, yes, you do get this precisely clear from minus 70 millivolts, as I said, to about plus 20 millivolt, a nice, this is called depolarization, a pulse. Because it's thin and narrow in the context of a lot of long-term recording, we see it as a spike. You can call it that, you can call it action potential or whatever, but it's the way information is encoded. What Hodgkin and Huxley wanted to know, like why, what's in there, and that's what they figured out, that during this period there's a big flow of sodium, because the cell membrane is permeable, conductive to sodium, and for rest of the time it's permeable to potassium. So this is what is called depolarization, and this is what is called repolarization, and essentially it's an exchange of sodium and potassium. So the way they did it was also amazing. What they said is that I must figure out a way, for example, to prove that it is sodium is important, which was a good guess. People knew before that, because after all, body is full of salt, water, we all knew. There was enough knowledge that inside the cell, you know, and outside there is a ratio of sodium and potassium. So people knew, Nernst law was available. But what they didn't know is how was the action potential produced, how was the flow was occurring. So how about if I block sodium? That's good, right? If I block sodium, what's left? Potassium. And if then I'll get a bump like this, and I'll be able to say, oh yes, this part of the action potential is due to potassium, because I blocked the sodium. If I block potassium and I get only this part of it, then I know it's sodium. How do you block sodium? Well, where do we find all these things? Some creature somewhere out there, some cobra, something has a poisonous.
Venom.
Yes, it's venom, exactly. Some snake venom, there's one called TTX, tetrodotoxin, and that one works by blocking sodium channels. So essentially, when you think of the snake, I don't know if it's cobra is the TTX, but that's what it's, something like that. It shuts down, basically that venom blocks that sodium channel activity. Suddenly no more depolarization, neurons cannot work, you will instantly paralyze, suffocate to death. That's probably one of those things first happens. Of course, eventually brain shuts down, everything shuts down. So it's superly poisonous because of that. So they found that, and again that knowledge might have present, but they had the understanding that why don't we apply TTX, block the sodium channel, figure out what the currents are. And same thing there with potassium, and that sort of opened up a line of thinking, and then how they understood that. So then that Nernst equation now can be expanded to cover a lot of different ions, potassium, sodium, chloride, to find a very balanced view. So there's a Goldman, you know, this is a Goldman, Hodgkin, you know, so later on he got another Nobel Prize later, but these are the people and the many others who contributed to this. So this is, I was telling, right, and for a while I was defending the biology is not like Newton's laws, but here we have nice, wonderful, precise equation, Nernst law, Goldman-Katz equation, some more other rigor has come in. So eventually we will get it, right, and not all of this will fit our simple math right now, like this is simple math, right, but it can be nonlinear, partial, and then eventually stochastic and quantum and whatnot. So the mathematics for some of the neuroscience may also in the long run emerge, but this is was very helpful. So now they can see that in reality how this looks like. Extracellular, lots of sodium, potassium, ions are flowing, and you see during this early part sodium permeability went up, and the later part sodium permeability went down, but potassium permeability went up and potassium started to accumulate. So what they saw, which is the most relevant interesting part, that it is an what is called all or none phenomena. Whatever low level of little current input, no response, but as soon as it becomes large, just large enough, you get this pulse, full action potential. So that is where all this, remember those dendrites and all the thing are shown, all little tiny voltages, lots of little but may not add up, but if there are enough of them, they add up and go above the threshold, boom, you get a spike and you get an output. So that is the way that all or none phenomenon works. So here is the picture of the squid, and this is a ganglion, so it is a nucleus of cells, but here is the giant axon. So this big axon, it looks like a tube, and instead of, to make life simpler to understand, they even replace the inside fluid with physiological salt solution. You can have concentration that you want inside, concentration you want outside, and see what the inside to outside flow is, and insert this wire to measure the potential in the cell, and that is what they found, that these are action potentials. So on. So I had shown you this circuit a little bit before. You don't need to understand it, but it captures sort of the things that we saw: sodium channel, potassium channel, all of that work together to produce the membrane potential. Resting is about -70, then depolarizes to go to -20. Some cells it is different. Sodium and potassium is just the start. People have discovered so many different ions and hundreds of different channels. So there is a lot of diversity, just like neurons, there are these big pyramidal neurons, but then there are smaller neurons and a lot of variety. So of course naturally there is a lot of variety, but simplistically it is this idea. And then added to that, you can have two types of synapses, excitatory or inhibitory. You can excite a neuron or inhibit a neuron. So let us bring a little bit of physics or engineering to the channels, because it seems like if I tell you it is a bubble or it is like a soap bubble and all that, it is true, but you do not fully get it, right, you like more rigor to that. So we can do this, right. So soap layer, these are totally insulating, so they do not matter, and then there are channels, so they open or close. So we can model the channels as conductances. So if we have two channels, we can put two of these resistors, inverse of resistance as conductance, and now you can have a conductive membrane which may be on or off, it is open or close. Okay. So now maybe it's, these are for those of you, I keep some of these in my notes because some people, my clinical medical people may not have done circuit. So this is the fundamental circuit tutorial of how to analyze circuit, don't worry about it. So now we can and these lipid layers can be modeled as a capacitor, right. So they are insulating, so they are capacitor, capacitors hold charge. So you can see, and this is how capacitor charges and discharges. When you charge or discharge a capacitor, this is the current you get. Then now you have a more accurate circuit, right. Lipid as a capacitance, channel as a resistance or conductance, and now you can see that if you apply a voltage across, or current, if you apply in this case a current, you are getting a change in membrane because capacitor is charging and then discharging. You got it? See, now it is sort of starting to make sense, that little bump we saw earlier. Do you see this? See, it is charging, right. It is charging to its threshold, it is charging to its threshold. That is because this is what it is. So, you know how elegant it is, sort of going back and forth between physiology and physics or circuits, you get this understanding. Yep, we are charging, and then of course it will discharge. You can put time constant and all that. So, instead of brain machine interface, let us call it cell machine interface. So, you can put a pipette or micro electrodes in it and measure all of these voltages. So, resting membrane potential, then this is your membrane, resistor and capacitor, and then charging. Okay, this is getting a little technical because nothing is perfect. So, there is always some leakage and it makes for some error in measurement. So, I think let us just ignore that for now. Okay, another Nobel Prize. So, before we go there, let us go back and review what Hodgkin and Huxley did, right. Everybody understand what they did, that they saw that squid axon served as a membrane. It has permeability to sodium and potassium ions, which go back and forth, which in turn produces this all or none action potential, all of that. That started to explain a lot of things about physiology of cells, and not just neurons, which is how it started, but heart cells, kidney cells. A lot of active cells work like the same way, just different ions, different little properties. But how do the ions go in and out, right? That part, so they speculated, they postulated, as they call it, that there must be channels, because ions have to go through something and they can't go through lipid. So, there must be channels, but they couldn't see it. There's, how do you know what is in there? So, they just speculated, and they did those equations. They found the certain ratio of conductance, and conductance in channel meant that certain property of channels must exist. And this was like late 40s or something. So, it took about 30 years, but then two people came about, Sackmann and Neher. They are German guys, I think, and Sackmann is a physiologist and Neher was a physicist, and they got together, say let us build some way to measure the channels. So, they came up with this idea of what is called patch clamp. Let me try to maybe go to this figure first. They said, okay, if there is a channel, I mean for people like us, if you think of that there is a membrane, there is a channel, our instinct would be, my instinct would have been to get things like some micro or nano electrode and push it in the channel or something like that. But that kind of does not really make sense. It does not prove anything. You can't force open a channel or something. So, how do I measure a current? I mean, I can't, and if it's a nanoscale channel, I cannot find it. So, their idea is kind of brilliant. They said, let us make a pipette, again pipette, little tiny glass tube, and bring it close to the cell. It does not really matter. I mean, this is pretty big actually. It can be about 1 or 2 microns, quite big. I mean, the scale is not correct. The channel is still nanoscale. But if I bring this pipette, I will trap 1 or more channels here. So, let me trap it. So, they said, okay, let us make this nice smooth pipette. So, they figured out, took a glass and learned how to polish it, so it will not puncture, because again cell is like a balloon, right. If you put a needle in a balloon, what happens? Pops. But if you insert it slowly, sometimes you can actually get the needle through and then balloon does not pop. But if you suck to the balloon, it will remain, right, it will pop out, right, you can pop out the balloon, you can suck it on it. So, roughly it was like this, that let us bring this glass pipette, and then soap tends to stick to glass, right. So, it creates a soap film. So, it makes the nice insulation soap film, and wonderfully now this little channel got trapped. It turns out that there are not too many channels. So, wonderful, it is easy to isolate a single channel, although sometimes if you get 2 of them you will see signal like this. So, let me explain that. So, as soon as when that happened, they started to see this current go in and out, open and close. There was a little quanta of ions going in and out through the channel. Wow. They discovered that there are actually ion channels, and like gatekeeper they are letting ions in and out like a digital gate, and it is stochastic. So, by the way, you see how random they are. Things at small scale are quantum, stochastic, it is pretty common. So, there is this, when you apply some voltage, the channels open up, quanta of sodium or potassium appropriately go in and out. They got Nobel Prize for that. What's called patch clamp technique, they call it patch because it's a microelectrode that patches to the cell to stick to the membrane. Clamp means you have to clamp it at a certain voltage to elicit proper current, V equal to IR. So, for different conductions you need different voltage, and then you suddenly observe this, and then now it makes sense that if there are two of them, then sometimes you will get twice as much current or thrice as much, then there were 3 channels. But most often you work with single channel to figure out what is the relationship between voltage and current. Okay. So, not only can you do it with the whole cell, but you can actually take a patch of membrane just like Hodgkin and Huxley took squid axon like a tube. These guys took you can pull out a membrane, you can just tear it off the cell. So, cell will die. Now, you are left with the membrane, which is this membrane with a channel in it, and now you can just study the pure property of the channel. It's a protein, it's a gate that opens it because the proteins opens and closes. Subsequently, there have been another Nobel Prize for people who figured out what a potassium channel looks like, and at Johns Hopkins there is a scientist who figured out there is a water channel, so he got a Nobel Prize for that too. I think they got it together, so water and potassium together, as I remember. So, because how does water get in and out? Okay, everybody take a deep breath. Shouldn't it have occurred to you how water goes in and out of cell? Did we all bother to stop and ask the question? Couldn't we have gotten the Nobel Prize? I mean, it's the same technique, right? And figure out how water goes in and out. That's the genius of people who get the Nobel Prize or luck and various things. There is a number of combination of things involved. And so, this is how these voltage clamp technique, as it is called, Hodgkin and Huxley discovered, and Sackman and Nair discovered this patch clamp technique, and they are very standard techniques or technology in physiology to understand the properties of what are called excitable cells, cells that responds to voltages, current, produces extra potential. So, these are heart cells, brain cells, sometimes some certain type of other kidney, and certainly muscle cells, okay? And that explains to us why muscles twitch, heart beats in a periodic manner, because it is the same thing. There is an action potential and there is a flow of sodium and potassium, and there are a lot of calcium channels are involved, which is what differentiates and so on. And now people are also finding that there are a lot of disorders, particularly in the heart, are related to this failure of these channels and channel proteins. A lot of drugs are just, you saw that TTX block sodium channel, it's one way to kill somebody, please do not try. But there are ways to. Yeah, so phenytoin, these are milder versions, so that when there is a hyperactive, it is a drug for epilepsy. Remember, epilepsy was a seizure, sort of indicates high excitability. So, phenytoin is a drug for epilepsy. It is not used anymore because it is kind of like it has too much reactivity, but it is a first order drug. Probably now India it might still be used because generic and cheap, so it can be, there are better drugs now, very expensive, because they also have reaction, because you are blocking channels, so you can have all kind of other issues. So, you see it can be for treatment. Okay, so maybe go a little bit further. How do you actually model it? How do you record it? So, what they did sort of like this is the patch pipette, and then a microwire is going in, and this is a circuit. For those of you know circuit, it is a current to voltage converter. So, any current from here in an op amp goes out and produces a voltage. Simple equations, the current is proportional to this, voltage is proportional to the current multiplied by the resistance to produce the voltage. So, now I can give a little tiny pulse of voltage to depolarize the cell and measure the currents. So, this is the patch clamp circuit model. While voltage clamp was a little bit more old fashioned way, where you put this tubular thing here, this is our Hodgkin-Huxley kind of thing, and then you apply, it's getting a little complicated, but you apply. So, there is one electrode here, the other electrode here. So, you measure the membrane voltage, but then say I want membrane voltage at minus 70 millivolt, that tells me which current is present. At 0 millivolt, then I depolarize it, I mean minus 50 millivolt, then I depolarize it and see what current it is, then I got it all the way to 0 and back. So, this voltage clamp circuit is a way to figure out ion channel current at different voltages. For again those of you trying to understand, it is very simple, you know, you are applying a voltage. So, this even a cell has a voltage, and membrane is resistance and capacitance. Rm membrane resistance, so membrane conductance, conductance is opposite of resistance, membrane capacitance, and then you measure it. This get a little bit complicated, so let us just move forward. I mean there are different ways of doing this. So, there are many different types of electrodes. There are sharp micro electrodes that go into the cell to measure voltage. There are these patch electrodes that measure currents, 1 picoampere to 10 nanoamperes, so very small currents. And there are metal microelectrodes. When did I last mention these metal microelectrodes? For what? Neurons in the brain, because this microwire I can stick it in the brain sitting outside the cells. You remember I was telling you about it, because brain cells are all packed and then shove a metal wire in there. Now you can record the potential of the neurons nearby. So, microelectrodes are the really important tools for neuroscience. Record from inside if you want to study fundamental properties, record from outside if you want to build brain machine interface or anything else. Okay? And we will talk about different design of microelectrode, all that later. So, now you know you now have tools, pipettes, you know, these are details of how to make pipette and so on. You know, you have to make it typically out of glass and then fill it with solution, and it's got resistance and currents. You have to, the other thing is that you use the silver chloride, it's a way to make a reference electrode. So, I think we can pass that, I will talk about later. So, now a little bit earlier, you remember I was talking about electrical engineers have modeled the axon and all of that. So, we are coming to that. There it is. This is that cable, as I was telling you. So you know, Hodgkin actually made it easy for us to think of an axon like a tube. I think if we had seen that biology picture, you know, it would have been kind of difficult because it showed axons seemed so small and 3-dimensional, all that. But Hodgkin actually kind of made it easy for us to see it as like a tubular structure. And now it's essentially the tube is membrane. It has inside and outside, and now we can model it, right. So, membrane is resistance and capacitance, and then there is some voltage that is produced across the membrane. Obviously, this cytoplasm which is inside the cell, of course it is a salt, right, it is some kind of conductive salt solution. So, it will have some resistance, and it is distributed. What this is called, it is a distributed model, which means there is not a single patch, but I mean, how do you model a continuous patch? So, well, let us break it up into little pieces and call one piece maybe 1 micron piece this, another micron, another micron. I mean, I can make it into a cable. That is why it is called a cable model. See, we can bring some physics, some precision to biology. Otherwise, it was looking like 3D wee stuff, but now we have some precision. And then if you stimulate without, if there were no channels, if I apply a voltage, okay, if I apply stimulus voltage, it will be, there will be a voltage and it will just decline, because the capacitor, right. So, it is a capacitive effect, and and there is a resistive effect. So, the essentially the voltage will degrade because it will be high in the beginning and slower, lower and lower and lower, because it is degrading, right, because of the resistance along the cable and capacitance along the cable. By the way, it goes in both the direction until in reality I want it to go in one direction later. So, we can see that, and now we can put some equations to that to fit these curves. That is reasonable. So, you know, solve this equation and say this is what is called space constant. So, how far before it goes below 37%. So, a lot of physics people did this kind of modeling for a few decades. Of course, back to myelin, right. So, remember myelin was that Schwann cell oligodendrocyte that coats or covers the axon with a tube inside. or if you look at an electron microscope, that's the highest resolution microscopy you can do, although that's not sort of true. There's now, it's generally true, but recently super resolution microscopy came out, so optically you can do that too. There was a Nobel Prize for that. Somebody keeping track of Nobel Prize I'm throwing around only today, okay? Tomorrow we'll be just doing simple stupid stuff. So here is the axon, this is the myelin, and it basically shows that the space constant improves tremendously because there is no leakage of current. So the current can travel very rapidly until it reaches the nodes of Ranvier, which is the name given to it, and then it jumps and jumps, and so it travels very quickly. So this is what that picture is showing, that you have these channels at nodes of Ranvier, and this is the insulation, and then it will jump from here to here to here. So this is what it is, point A, then point B, then point C. So it's a what's called propagating action potential. Action potential propagates along the nerve. Are we beginning to understand that, right? Brain, spinal cord, they used to have cell body, they have axon, but we didn't know like how does the message go. Now you know, right? First of all, the message starts in the cell, Hodgkin-Huxley, action potential, ion channel. Now you know it propagates. Either it propagates across the membrane or with myelination, jump, 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 it propagates. And then it forms a synapse and it jumps to the next cell. So now you know how the messages are coded in the brain or nervous system. Pretty good, right? I mean, it takes few Nobel Prizes to get there, but now the picture is clearer. Okay?
So in previous slide, RM stands for RM.
Membrane resistance, RM. M is always for membrane, so R is resistance of the membrane, CMC is the capacitance of the membrane. Now we can have it across the membrane, so that's the transverse across the membrane, and this is along the membrane, so this is a different name. So inside, because it's salt solution, you can draw the same as outside also. But that tells you that, right, if I apply voltage here, that there will be some charging and discharging, and then some portion of it will reach for further. So when it starts like this, it slowly declines, right, because the voltage will keep dropping here, right. So it starts with a high voltage, then some of it will drop here, some of it will drop here, will get smaller and smaller. So when you stimulate a nerve, at some distance it won't be effective, it will be effective in narrow. So remember all this dendritic connection network I had shown, they affect nearby, they don't affect at very long distance, because so you can stimulate a block nearby because of this cable property. And a lot of people do. If you give me a whole network, and you can write a full equations to model the whole network of this based on these. And of course in reality there are a lot of other factors much more non-linear than this. Okay, so I think we can take a little pause. Not that there is a deep thought, but I am happy to answer any questions before we go into neural network, where that people will be interested because more application oriented. Okay, so I also kind of take a deep breath, drink some water, say hello to your neighbor. If you need to step out for a few minutes, it's part of the same lecture, but I don't want to kind of go on and on, so take a little break. One big change I see compared to what I'm used to in US or in particular in Singapore, you guys are not big on using cell phone. So either cell phone is too costly, or they are blocking the message, or you are really good students who are very attentive. Otherwise, it's the most annoying thing. Today, you saw I was a little bit annoyed at you coming late. Now, normally it's like that people are looking at cell phone and checking emails and Facebook or whatever it is. So I think you are all good sincere students. Let me take a few minutes break, okay? Just 5-7 minutes, don't go for long, okay, if you have to go to bathroom break or just stretch. Because we'll take another break in about 15-20 minutes.
I had opportunities to face all kinds of technologies, right from missile technology to aeronautics to human capital management. My primary interest was to look after the human capital management and how well we can do that. And cognition as a matter had come to my mind when I was working there, because we had a very big organization. We had roughly 8,000 scientists and engineers who are working in DRDO with 52 laboratories, with more than 252 institutions throughout the country and outside the country as well. And about 32,000 people work in these institutions. And one of the major jobs that we do is to look after the cause of Indian Armed Forces. While working there, what I noticed is that the area which is not been taken due care of is the area of human cognition, which has become so important. Because much of my time has been on Siachen, on borders with China, and also subsea conditions, under the sea conditions, then in aerospace areas. Human being function everywhere, and particularly human being when they function in microenvironments like submarine, like some of the shelters where you cannot even maneuver more than what you can expect. Under such conditions, how human beings function. So that was my challenge. And we being a R&D organization, our job was to make sure that along with the technology, how we can bring technology to the support of the Indian Armed Forces. So I will simply share my experience rather than going by the things which is there in the book. And my topic is cognition, though it is very basic and general, keeping the heterogeneity of the audience in mind. I thought the audience must be very diverse type. They must have come from various disciplines, including our own engineering sciences department. So I kept it very general. Therefore, before I actually begin, I would like to submit that I would not enter into anything which is very technical, but I will certainly enter into certain things which we require tomorrow. That is, from the Ministry of Defense, they actually look for certain technologies which relate to cognition and some high-end technologies, and they are absolutely required, because more than an intercontinental ballistic missile, we are interested in empowering human being, because we are a manpower intensive country. Maybe we are also becoming technologically intensive, but what is an asset to us is manpower intensiveness. Can we make them a little more powerful? Can we bring little more capability into their day-to-day activity? That's what was my interest. So while speaking, I'll simply share some of my experiences. But let me tell you, a lot of us have been doing research, but probably none of us have done research in conditions where you stay at 20,000 feet, sustain the climate and the weather of -70-degree temperature, and understand what goes in your mind. Many a times we see that people develop various kinds of difficulties in those environments. Very few researches have been done in those areas. Simple cognitive failure, for example, is a very important issue. People fail to recognize things very easily under such circumstances. People have got difficulties in remembering. They find it very difficult to remember. They fail at times to remember their son's name. because they are in a condition where oxygen is very little. We have a lab at 18,000 feet, a full-fledged lab we can, and where cognitive researches are also being carried out. But I will not talk about anything which are militarily sensitive, but definitely I will tell where our people can continue doing research in those areas. So, to begin with, let me tell you that by cognitive science, what we understand that by cognition we believe that it's a representation of something we see, and we know through this representation the brain develops certain forms of transformation, and those transformations are also being done through certain computations in artificial intelligence, and the computed cognition is synthesized into some form of electronic devices which you call as implants. So, cognitive science has become important for us to augment our capability, and this is the way we try to understand cognitive science. And by cognitive science, the behavioral science part tells that what are those elements in cognition which are of more importance to all of us, that when we talk about how to register information, how do we process information, and how do we retrieve information, this whole process has got certain elements which we try to develop in the process when we try to cognize certain things. So, I will simply tell in quick time that how these things process. So, when we talk about the functional architecture, basically we talk about certain steps. The steps are like how we sense, how we accept energy, how we perceive, that is how we interpret energy. Then we do not interpret everything, how we attend to something. Then the question comes that when we attend to something, we also memorize those things. If we do not memorize, we need to learn every day. Therefore, based on memory, we learn certain things. When we learn certain things, we try to solve problems with the process of learning. When we try to solve problem, we find that intelligence plays a big role to play. And finally, we see the feeling component of it, the intent component of it, the emotion component of it also moderate our problem solving architecture. So, when we talk about basically cognition, we talk about all such steps, and we basically do research right from acceptance of energy to ultimately a decision when it is really taken. So, in behavioral sciences, each one of them takes 6 months of time to understand that how sensation takes place, how perception takes place, how attention takes place, how do we memorize things, how do we learn things, how do we solve problems, how intelligence works, and how our emotion, our feeling, our intent moderates our decision making. So, ultimately when you take a decision, a host of activities actually take place simultaneously. Lot of biological researches are also being done, lot of psychological researches are also being done in order to understand how they amalgamate, and finally we arrive at a decision making. What is important to us, very basic things I am trying to let you know, very basic, that there are certain methodologies we use, and most of these methodologies are known to all of us. They are either clinical method or experimental method. By experimental method, the most common methods we use are electrophysiological methods. We also use a lot of imaging techniques, that is functional MRI methods. We also have certain behavioral techniques, and we also have got computational methods. And the clinical part, of course, is based on the kind of impairment you have. We try to see that if there is a damage in a given brain, given side of the brain, or a given part of the brain, if it is either a focal damage or a non-focal damage, how does it hamper your day-to-day cognition. So, what is very important for us to understand the basic methodology, how do we study cognition. And as I said, when we talk about studying cognition, every method we found has its own disadvantage in order to enhance their capability. So, that is a big problem. So, I have identified cognition as a major area where most of the R&D is to take place, because there is a large gap going on between the technology development and human capability development. To sort it out, I visualized a roadmap for our activities in human capital development in armed forces. The roadmap is that as of now, through training you can augment certain capability, but from augmentation we have to go to automation, and from automation we have to go to autonomy. There are 3 stages we thought that we will go ahead with the technology development along with the human capability development, 3 stages. The stage 1, I decided that nothing is drawn from any book. These are all my personal experience I have scribbled here. This is, I decided that let there be a human assist system, which I call as a augmentation of human capability. The present capability, can we augment them with certain forms of training, which includes cognitive power and processing speed, which includes affective and effective skill variety, which includes inductive reasoning and intuitive decision making. We can train people to decision making, but the question is, when a human being takes the decision, he does not know whether he is taking the best decision, or he is taking the good decision, or he is taking the right decision. There are different kinds of decisions, and we can train them how to take right decision, how to take good decision, how to take best decision. We can train them, but that is up to the human capability. The existing capability can be utilized in that form, but from there we have to probably augment our level to human as a system, where automation comes into picture. That is, with lot of automation we would like to reinforce human capability, we would like to reproduce human capability, and we would like to replace human capability. And if we can do that through the process of automation, we would like to enhance our level to a level which we call system as a human. So, human assist system, human as system, and system as human, which you call as a complete autonomy. The idea is that we are interested in developing finally to go up to the level of system as a human, but to reach that, until and unless we understand cognition as a process, until and unless we synthesize that computed cognition into the system, will it function as a human being? It will not function as a human being. So, our ultimate aim, that who will do the extraordinary missions? The robots has to do the extraordinary mission, the autonomy has to bring in, who will perform without human intervention, and who will follow the rules of engagement, because when humans are involved, there are a lot of mistakes the soldiers make at times under the exigencies of a warfare. So, we are also interested that the warfare must be having the proper rules of engagement. So, rules of engagement, then performing something without human intervention, and doing extraordinary jobs, lot of extraordinary jobs. All kinds of warfare takes place, let me tell you. The biological warfare takes place, the chemical warfare takes place, the nuclear warfare takes place, where human beings cannot enter even. Under such conditions, who will perform the activity? Somebody has to perform the activity. So, our final goal or aim is to get certain form of autonomy where such kind of warfares could be fought. Therefore, our interest that we would like to unify technology with human cognition. That is, we will move from technology enabling cognition to cognition enabling technology. That is, human as system to system as human. And in order to do that, I have decided that if our people, people who are sitting here, who are really working towards that front, can move to 5 different steps. My principal aim here is to submit to you that we do not have much as of now. Can you contribute into these areas, and what are those areas? For example, can I reinforce human capability? How can I reinforce human capability? Can I give someone 360-degree vision? Now, giving 360-degree vision technologically is not a big problem. You can have a helmet with sensors integrated, and you can give 360-degree vision back and forth, no problem. But the point is that will a human being be able to consume 360-degree vision, synthesize it and be able to take a decision at that particular point of time? People will be unconscious if he is forced to do that. Point is, we are only hardwired for processing 180-degree, but is our brain capable of making 360-degree vision? Getting a helmet like 360-degree vision is not a big engineering challenge. What is a big engineering challenge is, can I get my brain ready with 360-degree vision? Can a human being be trained in that condition? Likewise, having night vision goggles or night vision devices, we call NVD, is a very huge difficulty, let me tell you. No warfare is fought at daytime, everything is fought at night. But do you think that all soldiers will go with the night vision devices? I don't know whether you have seen night vision devices or not. You can actually fix it up to a particular point, a particular, suppose 200 meter, you can fix it, but you cannot see what is happening below you, just beneath your eyesight. You cannot do that. So using a night vision devices and going for the warfare, it can only be utilized for vigilance purposes, not for war front. But can I get some kind of capability in human being who would be able to see during night? And it's possible. We have identified several people who are color blind, who have got mostly rod cells, no cone cells. They can do excellent night vision. They can do excellent night vision. I need to identify such people. We call them field independent people and people with color blindness. Now this is a challenge, it's a cognitive challenge for us. I want people who can see at night. So that's a cognition challenge. If somebody does research in that and finds what kind of vision, what kind of technology can actually enable us to see during night. Likewise, camouflage detection is a very big challenge for all of us. Camouflage detection, every Air Force pilot is supposed to do some kind of training in camouflage detection. In warfare, everything is camouflaged, everything. But it's a special cognitive ability that somebody is required. How do I train a person to be trained in camouflage detection? Is there a methodology available? There is no such methodology available as of now. Doing fundamental research and doing somewhere which is application oriented is something different. In fact, whenever we do research, we try to identify at what level we have been doing research. In cognitive research also, let me tell you, our attempt is not to bring new knowledge from no knowledge. So researches in cognition can be done at 7 stages. We can bring no knowledge to new knowledge. We can have new knowledge to theory building. We can have theory building to principles of application. We can have principles of application to discovery of solution. We can have discovery of solution to translation. We can have translation to production. And we can have production to end user integration. And we can have end user integration back to no knowledge to new knowledge to have Mark II of the product. So the challenge is when you talk about neurotechnology, the idea is to go back to the end user integration. Now most of us are interested in doing no knowledge to new knowledge and at best from new knowledge to theory building, and from theory building to principles of application some researches. Very few are in discovery of solutions and much less few are discovery of solution to translation. From translation to production, where lies the industry to participate in such kind of research? So until and unless you find that they can be productionized in a large number or they can be integrated with the common man's technology, I don't think any industry would be ready for that. So whenever we talk about some technology, in particular with the area of cognition or something which is related to this, it's a very huge challenge. So reinforcing human capability is a big challenge for us. Our next challenge is, how do I read human mind or human abilities? Question is it possible to have a EEG translator? Can I have a X-ray spec? Or can I have a subvocal thought analyzer where you think and we should be able to tell from the signal at the trachea or maybe at the vocal cord area we should be able to amplify those signals and find out the algorithm ultimately to understand whether the person is thinking in a given direction or not. So, interception research is badly important for us to read somebody else's mind without his intent. We also want to restore human capability, which includes all kinds. The transcranial magnetic stimulation, we all know, we do not want to. Cognitive retraining tools is a very important thing, because most of the retraining tools today in immersion technologies, most of the retraining tools are in the virtual reality areas. How do we train them? And also implant is a big area. I just read one article where our navigational capacity is being improved with certain implants in the areas of the brain. All kinds of capabilities. Well, cochlear implant we all know, this is a deficiency model, but we have a efficiency model also with the implant. The deficiency model is that you have a deficiency, you can get rid of it. The efficiency model is that you have got everything intact, can you enhance it further? So, even internets are being implanted in the brain. Some of the basic research is going on in some of the US laboratories. Point is that, can I restore such capabilities? Can I replace human capability? Like biometric algorithm. One of the major difficulty in our capability is that we cannot perceive the changes in the human face below 200 milliseconds, because our circuits take at least 180 milliseconds from one point to another point. Now, most of the actual human expressions take place actual, which is spontaneous, which is below 200 milliseconds. Can we actually detect anything which is happening at 40 milliseconds? Because then you will get to know what is the real emotion in him. After 180 millisecond or 200 millisecond, everything keeps changing, because you learn how to intensify it, de-intensify it, mask it, neutralize it, all kinds of display rule you understand when the face changes after 200 millisecond. So, if you have got a proper biometric algorithm, you can actually detect micromomentary expressions. We call it thin slices of behavior. The thin slices of behavior, if you can detect it, you can actually sort out lot of problems, and that is not a big issue. I think with a high speed camera, if you can detect it and an array of expressions in 1 second, if you have 20 different slides for that, you can actually find that after 200 millisecond things are changing. So, when it changes, in what form it is changing? It is a very huge human cognition problem. We want some answer from you. We also want to replicate human capability, which is can you have a UGV scanner which is fitted with certain sensors like this? Can we have robotic cognitions, and can we have humanoids? So, these are the roadmap we have created for ourselves in human cognition research. We want to read human capability, we want to reinforce human capability, we want to restore human capability, we want to replace human capability, and we want to replicate human capability. These are the challenges that we all have for tomorrow. Well, the various methods, I am not going to talk in great detail about it, because you are already aware of it. With the kind of lecture you are undergoing, I am not going to talk about all these things. These are very simple things for you, but let me tell you, the HBP, that is Human Brain Project, it is going on for several years. It is supposed to be over in 2023, and they are supposed to artificially construct a human brain. They are with the computing speed of exaflops. As far as I know, the latest status, it has not been able to create the rodent brain. It could not still do that. So, there are major difficulties in such kind of research. Such kind of supercomputers require, to replicate a human brain, requires very huge energy, and we do not know whether such kind of brain will ever pass through any kind of evolutionary process or not, because human brain process through certain developmental stages. Even if we develop such kind of brain with a computing speed of exaflops, will it pass through that kind of evolutionary mechanism? Will it be that plastic, and will it be able to cognize the environment? That is a major challenge. The cognition of the environment is a major challenge for the supercomputers. The other challenge is with the work of neuroimaging, the functional MRI. Lot of you are aware of it, that functional MRI researches are going on for several years. In fact, the laboratories that I had under my disposal, we had got all the facilities. I had about 3.5 Tesla magnets, as well as the positron emission tomography, the complete facilities were with us, and we have been trying to understand how a human brain functions. And how human brain functions is a mystery still. We do not know what really happens when a brain gets activated. And during functional MRI, we know whenever we think something, there is a neural fire. The moment there is a neural fire, there is depletion of oxygen. The moment depletion of oxygen takes place, nearby capillaries open. The capillary open, the blood supply comes in, and the magnetized and demagnetized blood is actually separated by the scanner, and they are blocked. So this is what is functional MRI. That's not a big issue. Issue is that what the brain is doing at that point of time, we do not know. If the activation is there, we do not know how do I correlate it with a mental function. We have found that in resting brain the activation is maximum. How do I explain it? How do I explain it? In resting brain, the brain is most active. How do I explain it? So there are many challenges, and these challenges are there that we do not know how many areas are participating in one mental activity like cognition. We do not know what the brain is actually doing when there is an activation. Is it an excitation or inhibition? We do not know. I mean, it's possible that a region can participate in a function, but not at all critical for expression. This is not at all critical. It may be activating, but it's not so critical at a given expression. So these are the different problems. My challenge, my suggestion to all of you, that if any one of you are interested in doing such kind of researches, you can actually apply for a project to DRDO, and it can be for metacognitive tools, it could be for decision making tools, it could be for thought controlled computing, it could be for thought insertion technologies, it could be mind reader. These are the bigger projects, big projects that we are thinking of tomorrow. I mean, as of now, almost no attempt is made in this country to come up to the level to have such kind of technologies where we can actually make some kind of dent in the capability of the human being. So I think X-ray spec subvocal speech analyzer is a very already current science has published certain articles on that. Thought insertion technology has got great clinical significance for obsessive compulsive patients. Actually, there is a very huge relevance for that. Thought controlled computing, decision making tools. In a net centric operation, you have to have a decision making tool. Of course, we know how the decision tools are built, but under a net centric condition, actually how does it work? We have got no idea as of now. In this institute, those who are from the institute, they know be knowing some of the efforts are being going on. I can tell you who are doing what kind of research. Cognitive profiling researches are going on in this department, electrical and ECE, and language processing is another area where we have been working in this institute. Micro expression is also being done, researches are going on in computer science department only. Cognitive motor behavior researches are also going on, speech analysis going on, cognitive arousal researches are going on, even 360-degree optical imaging and scalp IR thermography researches going on, cognitive enhancement researches going on, cognitive failure, simple cognitive failure researches going on, metacognitive researches going on, cognitive dysfunction researches going on, as well as cognitive restructuring researches going on. So in this institute, lot of researches are going on. What is needed today is to pull them together and come out with a good structure, so that cognition as a science, where behavioral science, neuroscience and computational science has a perfect marriage, that needs to be upgraded. And I think those who are interested in taking such kind of research forward will be of great importance, not only for R&D purposes, not only for eccentric kind of researches, but also for the Indian Armed Forces. I would like to stop here because I have another appointment in another 10-15 minutes. So, I would like to move, but if there is anything I can ask, I can answer for you, you may kindly ask me. I had to anyway, if there is anything.